Good morning or good evening, guys, whenever you're watching this. Today, there's only 3 classes left, and 2 of these classes are going to be spent talking about stramenopila, which is conveniently highlighted over here. Now, stramenopila, we've already looked at it a little bit. We've looked at the mycota earlier when we were looking at heterotrophic protists, but today we're going to look at the photosynthetic members of this group. And these are the organisms you typically associate with LJ. Some of them are ones that live in the ocean. When you go to the coast, these are the ones you see. But let's break it down. So we're going to spend the next 2 classes talking about these guys, and then the last class we're going to talk about rhizaria. And that's it. That's all the time we have this year. Okay, so in our framework of how many instances of secondary endosymbiosis has occurred, again, the nomenclature is different in this. These are the heterocotophyta is a different way to, the different name for the same group, but they are secondary endosymbionts. So what do I want you to know for this class this week? Well, I want you to know what are the main groups of stramenopila, and I want you to know what's really important to me is their ecological roles. Doesn't talk a lot about the ecology in the text, but I'm going to talk about the ecology that I think is important in this lecture. But read the text. There's not a lot to read, and I don't go into a lot of detail in this lecture, but please read the text. The readings for this week are chapter 15, only these sections though, diatoms, golden algae, and yellow-green algae. And the golden algae is really, really short. So there's not a lot of readings this week, so please read them because that's the material in your text is going to be covered on the quiz. I hope that's clear. Okay, stramenopila. These are the groups that we're looking at today. These are the common names and the Latin names. Let's start with, before we start with the diatoms, I mean, this is such a diverse group. They look completely different, right? You've got these giant kelp forests, and you've got these microscopic diatoms, and you have these things that look like green algae. And yet, regardless of the classification system, they all come out together, right? So they're a monophyletic group, even though they're wildly different. Let us start with, oh my goodness, where am I going with it? Why are they grouped together? Well, they're grouped together based on molecular data, for number one, but they all share this very prominent trait. They have special flagella. They have hairy flagella, which sounds kind of profane when I say it, but it's true. They have 2 flagella, which is common among all sorts of groups of protists, but one of them is very hairy and straw hairs, and these are swimming cell. They have a smooth flagellum. The group is called, as I just mentioned, heteroconts. Different flagella. They've got 2 flagella, so they've got 2 names. Okay, but they are monophyletic. Okay, let's get to the diatoms. I've been trying to get to the diatoms because what else? I think they're absolutely spectacularly beautiful. I mean, desmids were beautiful, right? Dinophytes are beautiful, but these are out of this world, literally out of this world. They look otherworldly. They're so beautiful. Now, right, we've seen other organisms that look like this, the haptophytes, the desmids, the dinoflagellates, the diatoms today, and we haven't talked about the radiolarians, the rhizaria yet, but there's all these microscopic single-celled organisms that are highly ornate, really like little miniature sculptures that are so beautiful, and they're really well preserved in the fossil record. But this is a really good example of convergent evolution. These things all have, other than the fact that they're all eukaryotes and descended from whatever that putative eukaryotic ancestor was, these guys aren't related to each other. This is a successful body shape in nature over the last, I don't know, couple of 100 millions of years, and it continues to be so. And it just happens to be spectacularly beautiful. Oh yeah, there's more foraminifera. We'll talk about those in 2 weeks. Okay, so Bacillophyceae, or diatoms, are crazy little eukaryotes with silica in their cell walls. And the fact that they have silica in them means they're hard. They preserve really well. They're crazy little eukaryotes for all sorts of reasons, but number one is their bodies are like 2 halves of a petri dish. Literally, They're overlapping lids, silica lids. They're made of opaline silica, highly ornamented, but it's like the craziest life form. And they have all this thousands and thousands of taxa, and they have these slits. They're not flagella. The thing about diatoms is they only have flagella. The male sex cells are the only things that have flagella, and yet they're aquatic and marine, and they stream. And it describes this in your text how they stream on an ooze of goo. But there's all sorts of different shapes, and like I said, I like to show you pictures because I think they're so pretty. Man, like they're just crazy. Don't they look alien? I just love looking at them. Okay, the star shape. So there's pinnate, which are the bilaterally symmetrical ones. Sorry. Here, let's talk about them here. There's the ones like this one that are pinnate, that have bilateral symmetry, and then there's the centric ones that are like spherical or round. And these two overlapping petri dish halves are called frustules. Frustules, like saying that, I don't know why. And I already told you about the flagellates in the male gametes. And I told you how they glide on goo that's exuded from the raphides, but you can read about that in your text, so you can't talk about everything. Okay, so diatom reproduction, how do they do it? If you're like a little petri dish, how do you reproduce yourself? Well, it's actually quite easy. You just split up the halves of the petri dish and you make a new half, and just repeatedly. But because the dishes are they're different sizes, right? If you think of a petri dish, there's a little side and a big side. If you split up and these both become the big size, then the one that was a little size is going to always give rise to slightly smaller cells. And this is just what happens. So at some point, you've got a population that's like 30% smaller than it started out as. And so these organisms have adapted to, once they get to be 30% as big, they have sex. And once they sexually reproduce, they regain their normal size. I told you they were crazy little eukaryotes. So this is what happens. Each daughter cell receives one frustule and then forms a new half. Some of them expand during cell wall formation and they can maintain the size, but some of them can't, and then they get progressively smaller. Just talked about that. Okay, so sex. How does a diatom have sex? How does a petri dish have sex? I mean, it's not easy. So these guys have gametic meiosis. Oogamous. There's also some diatoms that have isogamy though, anisogamy. But so the centrics are oogamous, which means, remember, big female cell, little motile sperm cells, and the isogamy, the pinnate diatoms have this, and this means the gametes are the same size. Okay, so here is an example of, so what happens? Well, you've got these diatoms, right? They're single celled. Some of them become female when it's time for sexual reproduction. Some of them become female, some of them become male cells. They undergo meiosis within the cell, and then they undergo mitosis in the case of sperm, and they produce a few sperm cells, or meiosis in the case, or one egg cell in the case of the female cells. What else do I want to say about that? Oh yeah, why are they gametic? I have asked this question. Why are they gametic? Which generation is multicellular? Because remember, part of the way you figure out what kind of meiosis or what kind of life cycle these organisms have is you figure out which one's the multicellular generation. But these guys don't have multicellular generation. So the only other way you can figure this out is, where is meiosis occurring? And meiosis is happening in the gametes, because the organism is only one cell big, so the gamete is the cell. The gametic meiosis. No multicellular generations. Okay, so what happens in sexual reproduction, right? Oh, sorry, I've skipped ahead. Well, in the male cells, right, they open up so that so that the sperm can actually swim out, and then the frustules in the female cells separate so that the sperm cells can penetrate it. But then those frustules from the male cell just sink to the bottom, and that's why we have huge deposits, silica deposits at the bottom of oceans, and get fossilized, because that's why. Okay, so the zygote then is the fertilized zygote that's within the female cell then goes on to produce new frustrals. And the frustrals of both the male and the female cells sink to the bottom. And this is what creates something that's called diatomaceous earth. You might see this in a gardening store. It's just very soft sedimentary rock, and it's made out of diatoms. That's why it's called diatomaceous earth. It's very abrasive. You can polish it, it's used in filters, all sorts of industrial applications. Yeah, if you actually take some, if you buy some in the gardening store and look at it under a microscope, you can actually see the residue of the old diatoms. It's just diatoms. And they're used all over the place, pet care, pet food, bed bugs, cat litter, all sorts of things. This is also a problem because there's this place in Kamloops, near Kamloops, Red Lake Earth, which is this fantastic Miocene deposit, about 12 million years old, full of crazy, crazy marine organisms. But it's diatomaceous, so it's been harvested for forever. There's BC free entry mining laws have allowed the strip mining of this family farm. I mean, regardless of what the farm owner wants, if there's deposits there, they can be mined, and it's become a kitty litter mine. I kid you not. These fantastic, valuable fossil beds are kitty litter mines. Now, I don't know. I think there was a court injunction last year to stop this because it's an abomination. I haven't checked it out. Anyways. Okay, what about the ecological role of diatoms, right? That was not exactly an ecological role, but so phytoplankton, right? We've talked about that a bit. It's less than 2% of the Earth's total living biomass, and yet it's responsible for 50% of the net primary productivity. Okay, you've heard these stats before. But what you might not know is 25% of that 50% is due just to diatoms, these crazy little eukaryotes. These crazy little photosynthetic eukaryotes that look like jewels, alien jewels, are responsible for a quarter of the phytoplankton productivity. I mean, that's pretty fantastic. These guys are really important in the climate cycle, in the carbon cycle. They are, and because their shells last so freaking long, they are really important long-term storage for carbon. Really important little critters. Okay, so they're really important in the marine food web. That's implied because they're so important for productivity. So I don't know if you are aware, but there's been some movement over the last 10 years to boost the productivity of the world's oceans in order to combat climate change and also to restore fish stocks. In theory, it sounds good. So there's been some experiments where people have fertilized the ocean with iron, because iron is a limiting nutrient in the ocean. And the thought is you put iron in, you stimulate phytoplankton productivity, you stimulate photosynthesis, there's more energy for the food chain, and ultimately you sequester more carbon dioxide from the atmosphere and it stays into the ocean. However, the other option is there could just be a ginormous algal bloom and the food web falls apart and carbon ends up being liberated. Not really a good idea. So what happens? There are people fertilizing the ocean. Well, there's been some experiments and they consistently show that intentional iron addition, so ocean fertilization, results in phytoplankton blooms. So this is not what we want. This is not going to improve carbon sequestration or help the food web. Experiments haven't determined if the carbon is transferred and sequestered in the deep sea or recycled into the food chain at all. So it's completely up in the air, but there is some example where it's just not helpful at all. So in 2012 in Haida Gwaii, so this is a local experiment, an American entrepreneur convinced a Haida Nation village to fertilize the ocean in order to boost their salmon populations and also earn carbon credits. It sounds fantastic. So they went ahead with this, but the news didn't get broken until they had already dumped 100 tons of iron sulfate into the open ocean. I mean, how could that even be legal? Who owns the ocean? And this is what happened. There were massive algal blooms. So this is where, down here, this is where they fertilize, and yet there's this massive area of algal blooms. So it was an unmitigated disaster. I guess the point of this is diatoms don't need your stupid iron, because diatoms are especially uniquely and exquisitely adapted to grow under iron limited conditions. It's always been the case, and yet they're still super productive. So giving them iron isn't going to help their productivity, because they have very, very low iron requirements. They've really edited their proteome, gotten rid of iron demanding proteins, so they don't need a lot of iron to begin with. And they can perform luxury uptake of iron. So when iron is available, they can store it and use it later. And also they use photorhodopsin to generate ATP when iron is low. So they don't necessarily need it to be productive if there is a temporary deficit of iron. They don't need it. Fertilizing is not going to help it. And countries are starting to, this is 2008, I said they're starting to put the moratorium on ocean fertilization, and yet it's not true. A new company in Canada here, a Canadian company, is trying it in Chile. People think if they can make money off of it, they're going to try it. So that's why it's so hard. This globalized economy we have, how do you control what happens to the ocean? I don't know. Okay, so that was Bacillariae Fayeaceae, or diatoms, just a little snapshot of that. I want to talk about golden algae and yellow-green algae. I mean, here's the pictures again. Your book doesn't really talk about Chrysophaceae, golden algae, very much. I think there's a paragraph, and so I'm not going to spend a lot of time on it. They're unicellular. They can be colonial. They live in fresh water. They're gold because they have a large amount of fucoxanthin, the pigment. And here it is, fucoxanthin. They're strange little organisms because they can be, they have resting cysts, which make really good fossils because they're silicalized. And an amoeba emerges from the cyst when conditions approve. So they're crazy, and they can be colonial, right? Here's an example of colonial organism. That's about, and oh, there's a variety in cell shapes, right? Again, they're quite beautiful. Colonialism is really common, and they can be covered with these silica scales. So also there, and you can see the flagella sticking out. Very beautiful, very exquisite little creatures. But I can't talk about everything, so that's all I'm going to talk about with these guys. Okay, so let's go to yellow-green algae, and that's the last thing I'm going to talk about this week. Xanthophyllaceae. There's only about 600 species of these guys. They live in freshwater, marine, and soil. I don't know why I forgot marine there, because they are marine. No fucoxanthin. They look green because chlorophyll is exposed. So they're yellow-green algae. The reason, what I want you to take home about these guys is their role in the ecosystem, Arctic ecosystems. You probably know that Arctic sea ice is in dire straits, and almost every day there's something in the news. Arctic's strongest sea ice breaks up for the first time on record. Right, if you look at the sea ice volume from 1979 to 2018, and the y-axis is how many thousands of square kilometers are occupied by ice. This is 2018 down there. I mean, every year it goes lower and lower and lower and lower, which is a problem, right? I mean, I'm sure you've heard about the polar bears. They need the sea ice. They hunt on the sea ice in the summer. They are marine mammals, and without sea ice, they just can't survive. So this is a problem, not only for the polar bears though, but because the basis of the food web depends on sea ice too. So you can see this sheet of sea ice, and it's kind of colored green. It's because algae are colonizing the bottom of that sea ice. And that algae feeds copepods and other arthropods that are then eaten by the fish, that are eaten by the mammals, and that's how the food chain works. So these sea ice algae are really, really important. It's called the sympagic environment, and that's where water is mostly frozen and happens in glaciers or ice caps. There's 2,500 million kilometers squared of this. Well, there was when I wrote this. Who knows if it's changed. And 15% of oceans are covered by sea ice at some point in the year. And when they are, they're colonized on the underside by blue-green algae. So if you did a core, you could, so here's a core, you could see the bottom, it's just covered, matted with algae. So these conditions are really weird, right? They're low light, they're very high salt because of all the freezing water. So it's very high saline, and it's the basis of the food chain in polar waters. And up to 60% of the net primary productivity is due to these sea ice algae. So really, really, really important. Krill grazing on the sea ice is the next step up in the food web, and these guys feed all sorts of creatures. But the algae that live in these environments, they're unique. They don't live anywhere else. They're specially adapted to live on sea ice. And they're different than the ones that live right below them on the water column. So they're unique, which and here's some pictures of them because they're also really pretty. The question is, if the sea ice disappears, even if we're able to stop and reverse climate change and sea ice comes back, what happens to the algae that used to live under the sea ice? Because once the sea ice melts, they're the inoculum for all other sea ice. If we lose the sea ice, we kind of lose the algae. And if we lose the algae, these ecosystems are going to change irrevocably. Once again, these unintended consequences of climate change that we can't even predict, and it kind of hurts your brain to think of them. This is a study that just showed that algae from open water aren't colonizing the sea ice. So we need sea ice to populate next year's sea ice. It's kind of sad. I always end these talks really depressed. I'm sorry. Okay, that's it for today. I have got to go, and I will see you on Tuesday. And for the group who is going to present next week, we're going to meet Tuesday at 2:45. So message me in Canvas so that we can set that up. All right, have a good weekend.